The emerging field of nanotechnology has a lot of potential. For the past 2 decades, the field has seen a surge in interest and development. But what is nanotechnology? Nanotechnology is a field that manipulates matter on the nanoscale. Think microscopic, but 1,000 times smaller, the atomic level. The width of a hair seems huge compared to the typical nanoparticle. These miniature materials usually range from 1 to 100 nanometers, and more than 1,000 of these could fit across the width of a hair. The concept of nanotechnology was first popularized by Richard Feynman in 1959, and in his words, nanotechnology is making machine tools with the use of ever smaller machine tools. These nanomaterials have modern applications in medicine today, a notable example being the drug delivery system. Using the knowledge acquired from the biochemistry unit, we can analyze the structure of the drug delivery system. For example, in the liposome drug delivery system, the nanoparticle liposome encases a drug within its amphipathic phospholipid bilayer. The outermost layer of the nanomaterial mimics that of the cell membrane that has a phospholipid bilayer and peripheral biomolecules that, in our fluid mosaic model, were glycoproteins, glycolipids, and carbohydrate chains. Similarly, our nanomaterial has polyethylene glycol and other peripheral structures that help to stabilize the drug and deliver it to the target cell. In the course, we also looked at how materials pass through the cell membrane. And in the liposome drug delivery system, once the liposome reaches the target cell, it passes into the corrupted cell, like a cancer cell, for example. And it does this by doing receptor-mediated endocytosis. In the course, we also observed how hormones and other materials can use this form of vesicle-active transport into cells and locate them with peripheral receptors. In the same way, the nanovesicle attaches its PEG strands, which helps to locate and bind to the cell membrane. After it enters the cell, the nanoparticle exterior ruptures and the drug is dispersed within the cell. This is used for diseases like cancer, since it is a more controlled procedure that doesn't target healthy cells, and it's less expensive than a chemotherapy or radiation therapy. With innovative nanoparticles, scientists are in the process of building nanotechnology that can manipulate individual DNA genes and alter the way that a gene is expressed. This idea has just been developed in theory, but once it happens, physicians would be able to perform cell surgery. In theory, a chromalocyte would extract defective genes from a cell and insert digitally corrected chromosome sets. So basically, brand new genes. And we're not far from bringing this idea to life. In a study at Harvard Medical School, they were able to engineer medical nanorobots with molecules containing instructions that can make cells behave in a particular way. Think of genetic diseases that could be fixed using this new technology, like Huntington's, sickle cell disease, and much more. Researchers at the University of South Florida Health and Morsani College of Medicine have devised a way to help patients post-angioplasty to prevent the re-narrowing of arteries after the procedure. These researchers combine a non-toxic peptide with synthetic mRNA that codes for the protein P27. This protein inhibits cell growth. Within this mixture is a microRNA switch. This is a gene regulator, and it ensures that the synthetic mRNA codes for the P27 protein in target smooth muscle cells to inhibit cell growth, but it allosterically binds to the synthetic RNA when present in non-targeted endothelial cells to prevent the translation of P27 protein so these cells can divide and heal. Diabetes is a chronic condition that affects more than 2 million Canadians. Having diabetes means there is a lack of or an improper response to insulin, which breaks down glucose, and this leads to a glucose imbalance, offsetting homeostasis. The current treatment for diabetes is to inject insulin in the body daily to maintain glucose levels. But researchers at North Carolina State University have developed a nanoparticle system that can maintain glucose levels for a week. This nanonetwork is an injectable mixture of nanoparticles, insulin-modified dextran, and glucose oxidase enzymes. Once high glucose levels are detected in the body, the enzymes break down glucose into gluconic acid, which breaks down the dextran into insulin, which accurately maintains normal glucose levels, reinforcing balance or homeostasis. Efforts to bring this treatment to human trials are still underway. When the body sustains an internal injury, it works hard to reduce the effect of the harmful stimulus and works towards homeostasis. But for people who sustain traumatic injuries that cause internal bleeding from car crashes, sports, or even in the military, it could be life-threatening. As the central nervous system sends blood platelets to the site of injury to repair the skin, natural platelets might not be enough to curb the injury. Biomedical engineers at Case Western Reserve University noticed this issue and developed nanoparticle-tailored blood platelets that aid the body's natural platelets to create healthy blood clots and increase the rate of survival for traumatic injury patients. With all innovations comes limitations, and for nanotechnology, that lies in the unknown risks of using it. Though many back the production of nanotechnology-based tools in medicine, others fear the degree of toxic effects it could have on the body. Other arguments against it state that the production of non-naturally occurring nanoparticles can have drastic effects on the environment. But those who stand behind nanotechnology and its limitless potential believe it has the potential to better mitigate or even terminate diseases in medicine.